All right, let's dive right in. We're about to unlock the secrets of probably the most amazing instruction manual in the entire universe, your DNA. Seriously, it's the code that literally builds and runs everything that's alive. But that brings up a huge question, right?
I mean, how on earth does a single, tiny cell contain all the instructions needed to build a whole person? It sounds kind of impossible when you think about it. Well, that's exactly the mystery we're going to unravel together.
So here's our game plan. We'll kick things off with the big picture, the body's blueprint. Then, we'll zoom right in to see how DNA is coiled up into chromosomes and what genes and alleles are. After that, we'll see how a protein actually gets made, look at what happens when the code changes, and finally, we'll talk about why all of this really matters to you.
Okay, first up, the body's blueprint. Before we can get into the nitty-gritty, we got to understand the entire instruction book that it's all written in.
And that complete instruction book? It has a name, the genome. This isn't just one recipe. We're talking about the whole library, every single piece of information your body needs to grow and function. You can think of it as the master copy of you.
And get this is wild. If you could take the DNA from just one of your cells and stretch it out, it would be over 2 meters long. So how in the world does all of that information fit inside a nucleus you can't even see? Well, it all comes down to some seriously clever organization.
Which brings us to our second section, chromosomes. This is how that massive library of information gets organized into volumes you can actually manage.
So the secret is all in the packaging. That super long 2-meter strand of DNA is coiled up incredibly tightly, over and over again, into these structures we call chromosomes. These are basically the individual books in your genetic library. They keep everything neat, tidy, and easy to find.
Now for us humans, this library is neatly organized into 46 of these books, or chromosomes. They're arranged in 23 pairs. And here's the cool part. You get one copy of each book from each parent. That 23rd pair, well, that's the famous one, the sex chromosomes that determine biological sex, XX for female and XY for male. Okay, so we've gone from the whole library, that's the genome, to the individual books, the chromosomes. Now let's zoom in even closer and start reading the actual recipes inside. So a gene is just a small section of DNA on a chromosome. It's like a very specific set of instructions for building just one thing, a protein. And trust me, these proteins are the real workhorses of your body. They do, well, pretty much everything, from carrying oxygen in your blood to making your muscles contract. Your genes hold the codes for tens of thousands of these different proteins. And this is the perfect way to think about it. If the genome is the whole cookbook, then a chromosome is a chapter, say soups, and a gene is the single recipe for something specific, like tomato soup.
But you know how recipes can have different variations, right? Your grandma's cookie recipe might have a variation that calls for dark chocolate instead of milk chocolate. Well, in genetics, these little variations are called alleles. They're just different versions of the very same gene, and they can lead to different traits. Now remember, you get one allele from each parent, so you might end up with 2 identical versions, say 2 alleles for blue eyes. We call that homozygous. Or you could get 2 different versions, like one for brown eyes and one for blue eyes. That's called heterozygous. And this combination is what ultimately decides which trait you actually see.
So we know what the code is, we know where the recipes are, but how does the cell actually read the recipe and, cook the dish? This section is all about turning that blueprint into the real building blocks.
See, the master DNA cookbook is way too important to risk leaving the safety of the nucleus. It's like a priceless manuscript locked in a library. So the cell makes a temporary, disposable copy of just the one recipe it needs. This copy is called messenger RNA, or mRNA. It's single-stranded so it can slip out of the nucleus, and it uses a slightly different chemical letter, uracil instead of thymine.
The first step in this process is called transcription. Just think of it like photocopying a page from that cookbook. The DNA unzips right at the gene you need, an enzyme runs along one side, and it builds that matching mRNA copy. Once the copy is made, it peels off and heads out of the nucleus, on its way to the cell's kitchen.
This second step, translation, is where the real cooking happens. The mRNA copy hooks up to a ribosome, which acts like the chef. The ribosome reads the recipe's code 3 letters at a time. We call these codons. And for each codon, another molecule, called transfer RNA, brings over the exact right ingredient, an amino acid. The ribosome then links all these amino acids together in a long chain, which then folds up into a perfectly formed finished protein. Voila!
This whole process is amazingly precise. But what happens if there's a typo in the cookbook? Well, that's what this next section is all about, genetic mutations.
A typo in the DNA code is called a mutation. It's a permanent change in that sequence of letters. Sometimes they just happen, but exposure to things like certain chemicals or radiation can definitely increase the risk.
So let's look at a really simple example. Here's a tiny piece of an original correct DNA code. When the cell reads this, it builds the protein it's supposed to. No problem.
But look here, see that? One of the letters, a T, has been swapped for a C. That's a mutation, and even a tiny one-letter change like this can be a huge deal. It can change a three-letter codon, which means the wrong amino acid gets added to the chain. This can mess up the protein's final shape and stop it from working correctly, or at all. A devastating disease like sickle cell anemia, for instance, is caused by just one single-letter change in one gene.
But not all typos have the same effect. A substitution just swaps one letter for another. Sometimes that changes an amino acid, sometimes it doesn't. But insertion or deletion, whew, those are often way more serious. When you add or remove a letter, you shift the whole reading frame. It's like deleting a letter in a sentence and making every three-letter word after it complete gibberish. It can scramble the rest of the protein completely.
Okay, we've gone from the entire genome all the way down to a single letter change. So now let's zoom all the way back out and ask the big question. Why does any of this actually matter in the real world?
You know, understanding our own genome has been nothing short of revolutionary. It lets scientists pinpoint specific genes, like the BRCA genes, that are linked to a higher risk for diseases like breast cancer. This knowledge is helping us develop smarter, more effective medical treatments. It's leading to personalized medicine, where doctors can choose the best drug for you based on your unique genetic code. And it even lets us look back thousands of years into the past, tracing how our human ancestors spread out across the globe. It's incredible stuff.
So, for the first time in human history, we can read our own instruction manual. The code of life isn't a total mystery anymore, and that leaves us with a pretty deep question, doesn't it? If we can finally read our own book, what chapter are we going to write next? Thanks so much for joining me.